Our next speaker is Marbelys Pino from the University of Miami. She's going to tell us about Cdc42 dynamics and guess what? Microtubules again.
Thank you so much for that kind introduction. And I want to thank the organizers for allowing me to speak today and share some of my work. In the Verde Lab, we're interested in understanding the signaling pathways regulating morphology as well as polarized cell growth. We use Pombe Fission yeast as a model system because these cells maintain a cylindrical shape and they only grow from the cell ends while maintaining a constant length. The activation of growth at both ends, it doesn't happen immediately. Initially after cell division, growth only occurs at the old end, and not until the cells reaches a length of approximately 9 micrometers in length, growth at the second end is activated in a process that we call new end takeoff or needle. When you have mutations in proteins that are important for the regulation of Cdc42 or morphogenesis, sometimes this pattern of growth is altered, and as a result, we fail to activate growth on the new end. The example you're seeing here is mutations in the polarity factors T1 and T4, and these are polarity factors distributed to the cell tips by microtubules. And when you don't have these polarity factors, the cells fail to activate growth at the second end, resulting in monopolar growth. Oh, sorry. So today I wanted to talk to you about how the microtubule cytoskeleton and the NDR kinase ORB6 regulates the activation of Cdc42, and in that manner the cells regulates bipolar growth activation, meaning the growth activation at the second end. Polarized growth in Pombe is being linked to the activation of small GTPase Cdc42. And we show in our lab that the levels of active Cdc42 oscillate between the two cell ends, and we can visualize the levels of active Cdc42 looking at the bioreporter creep GFP. As you can see, these oscillations are anti-correlated. What I mean by that is that as the levels of active Cdc42 increase on one end, the levels at the second tip decrease. And this is tightly regulated with the activation of the growth at the second end, because not until the levels of active Cdc42 at the new end pass a certain threshold, growth is not activated. The small GTPase Cdc42 cycles between an active and inactive state, some like you might remember. And in Pombe, 2 families of proteins regulate Cdc42 activation. The ones that promote Cdc42 activation, 2 of them has been described. It's SCD1, which is an ortholog of Cerevicia Cdc24, and Gefl, which is an ortholog of mammalian Cdc42 tuba. I'm sorry about that. I keep going backwards. I don't know why. So I wanted to show you that one mechanism by which the cell it's regulating the activation of Cdc42 at the cell tips is through the negative regulation of one of its activators, Gefl, the Cdc42 Gefl. And one way in which we limit the amount of Gefl at the cell tips is by the phosphorylation of Gefl by NDR kinase ORB6. Phosphorylation of Gefl by ORB6 creates a 14-3-3 binding site and promotes Gefl interaction with 14-3-3 protein Rad24. And in this manner, we limit the amount of active Gef at the cell membrane and resulting in lower levels of active Cdc42. In a mutation in which you substitute the serine at the phosphorylation site in Gefl by an alanine, and in this protein, it cannot be longer phosphorylated by ORB6, and as a result, it cannot interact with 14-3-3 protein Rad24. And in these cells, we see an increased localization of the Gef at the cell tips and higher levels of active Cdc42. And because of this, we see that the cells have a wider cell diameter, and bipolar growth activation occurs earlier in the cell cycle of the cell. suggesting that the cell, by limiting the amount of, modulating the amount of CDC42, the cell can activate bipolar growth activation. And not until the cell, we have enough active CDC42, we are able to support growth at both of the cell tips. So currently, we do not know what mechanism in the cell we can locally activate the activation of Gefl, promoting activation of CDC42. Specifically, how we can negative regulate the phosphorylation of Gefl1 by ORB6. And today, hopefully, I'll convince you that the microtubule cytoskeleton plays an essential role in this process. Microtubules is very important for the establishment of cell morphogenesis in Pombe as well as in other systems. And unfortunately, tonight, I don't have time to go over all the work of many different amazing groups to describe this, to support this cartoon. But if you can please take my word for it, that one mechanism by which the microtubules promotes the activation of CDC42 and Pombe is by delivering polarity factors T1 and T4 to the cell tips. And in this way, the microtubules mark the cell tips for growth. Once at the cell tips, the microtubule polarity markers T4 and T1, they recruit other effectors important for polarized growth. And one of these effectors is the phosphatase 1, as well as forming and Gefl1. A great study by the Martin group showed that when you artificially move the T complex to the cell sites, you can recruit Gefl1 as well as phosphatase 1s to the cell sites and promote CDC42 activation in areas that normally would not occur. However, the mechanism by which T4 promotes Gefl1 mediated CDC42 activation is currently unknown. We showed in our lab that the microtubule cytoskeleton is essential for this accumulation of active CDC42 at the second end. And when you treat cells with the microtubule destabilizing agent, MBC, we failed to accumulate active CDC42, again visualized here with the CRIP-GFP, and the microtubules are in red. When you treat cells with MBC, we did not detect significant accumulation of active CDC42 at the second end. And in these cells, we see that the cells grow in a monopolar fashion. But it's very interesting that when you remove the negative regulation of Gefl1 by ORB6, by expressing the Gefl1 S122A mutant, we're able to bypass the microtubule requirement for accumulation of active CDC42 at the second end, suggesting that maybe that once you don't have the negative regulation of ORB6, in this case, the microtubule is dispensable for this bipolar growth activation. In a different experiment, we looked at bipolar growth activation in a mutant of T4, one of the microtubule-dependent polarity factors. And this mutant, the V223A mutant, fails to recruit the phosphatase 1 to the cell tips. Once you express the V223A, the cells usually high percentage of cells are monopolar, and Gefl1 is localized only at one of the cell tips, suggesting that this is important for not only symmetric localization of Gefl1, thus promoting activation of CDC42. In these cells, when you express the Gefl1 S122A, so thus removing the negative regulation by ORB6, we're able to suppress this defect and having bipolar growth activation as well as symmetric localization of Gefl1s at the both tips. So this results allow us to formulate this hypothesis that maybe one mechanism by which the microtubule cytoskeleton promotes bipolar growth and CDC42 activation is by opposing the negative regulation of Gefl1 by ORB6. To test this idea, we created an antibody specific for phosphorylated Gefl1. I have to tell you that there's only one NDR phosphorylation site in Gefl1, so allow us to ask this question with an easier way instead of you have multiple sites. When we look using our phospho-specific antibody in our Gefl1 S122A strain, we failed to detect any signal suggesting it is specific for this phosphorylation site and is ORB6 mediated. and in a different strain that is sensitive to inhibition by the kinase 1-MPP1. And this kinase reduces the activity of ORB6, an analog-sensitive form of ORB6. As you can see, through time, when you treat cells with 1-MPP, the levels of phosphorylated Gefl are reduced, suggesting that this antibody can be also a great tool to monitor the activity of ORB6 in cells. So using this antibody, then we went to test the idea, what was the effect of the T4, the microtubule-dependent complex T4, in the regulation of Gefl phosphorylation. And as you can see, when you overexpress T4, we see a reduce in the levels of phosphorylated Gefl, as we expected. I also told you that the phosphorylation of Gefl promotes its interaction with 1433 protein RAD24. So we decided to test if by overexpressing T4, we can see changes in the interaction of Gefl with RAD24, which is a 1433 protein. And so you can see, we saw a decrease in the levels of interaction of Gefl with 1433 protein RAD24 when we overexpress T4, suggesting that one of the purposes of the complex might be opposing this NDR-mediated phosphorylation of Gefl, promoting Cdc42 activation. So in summary, we think that one way in which the cells regulate the activation of growth at the new end is by opposing the negative regulation of ORB6. And as a result, promoting the accumulation of Gefl at the second tip to allow enough accumulation of active Cdc42 in these tips to support the activation of growth. And with that, again, I would like to thank you all for allowing me to speak today. And our special thanks to my mentor, Dr. Verde, as well as members of my lab, Elise and Chen, have been very supportive, as well as Dr. Das, which is a former postdoc in Dr. Verde's lab. She's now in Tennessee, as well as Dr. Wiley. I would like to give a special thanks to the NIH for supporting this work, as well as the JSA for a generous grant to allow me to be here today. And I will thank you so much.
We have time for questions, and thanks, GSA. She had to drive all the way here. You know.
I have a question. So maybe I just missed it too. So is the dephosphorylation of Gefl when you add the inhibitor of ORB6, analog sensitive, is that dependent on T4?
I haven't tested that, but that's a very good question.
Okay. Yeah,
so that will be something very interesting to do, yes.
Because I wonder, you're postulating that you're negatively regulating ORB6 locally, but it could also be just that you're dephosphorylating Gefl.
Yes. Right now, as the model that we have now, we cannot distinguish if T4 is important for directly affecting the phosphorylation state of Gefl, or it's affecting ORB6 activity, or it's affecting maybe other phosphatase that because there's other phosphatases that get targeted by this complex. So that's a very good question, and this is into the to-do list.
Any more questions? Okay. Thank you, Marbelys.
Thank you so much.